Good day, students. I am Comrade Isaac N. Igbawua of Microbiology department. I am going to take you on MCB222, which is staining and microbiological techniques. The course is divided into several topics, which is contents in this case, 6 of them in all. And in the arrangement, the topics have been broken down into weeks. So as we go through, each time we meet, we see how we can treat, and where we stop, we continue in the next one. And so in this week 1, we are going to consider introduction to microscopy. Introduction to microscopy. And we have a look at the principle of microscopy, the magnification of a microscope, the application of a microscope, and as we go on, you will understand as we proceed. Principles and application of microscopy. As a way of introduction, microscopy is the technical field of using microscopes, which are indispensable instruments in the microbiological laboratory, to view objects and areas of objects which are structural components that cannot be seen with the naked eye. In other words, microscopy involves the use of microscope in examining objects that cannot be seen without an aided eye, for the singular fact that they are smaller than what the ordinary eye can see. There are 3 well-known branches of microscopy, which has to do with the optical, the electron, and the scanning probe microscopy, along with the emerging field of X-ray microscopy. It involves the interaction of the electromagnetic radiation, if it is the optical type, or the use of electron beams with the specimen and the collection of the scattered signal to create an image. In other words, either the electromagnetic radiation of the light rays or the electron beams from the electron microscope, we create an image having passed through the specimen of the object under view to create an enlarged image. During the process, there will be an enlargement of the specimen by use of lenses and the resolution of fine details by way of adjustment and illumination, which has to do with the focusing also of the instrument microscope in this case. Now, a microscope as a principle enlarges the view of an object by enlarging it twice using the objective and the ocular lenses if you are using the optical type. And this is achieved through several mechanisms. as outlined here. 1. Light from below a specimen which is on the stage will be made to illuminate the specimen on the stage. And number 2, an objective lens which is placed very close to the specimen produces a real image of the specimen. Now the image is then created somewhere between the two lenses. Now the ocular lens looks at that real image initially produced, which is already enlarged, and then creates a visual image of it that is many times magnified. This image is created far away behind the actual objective. Now this final image is inverted to the original object. When the light passes through the specimen and the two lenses to reach the eye, the loss of light can be negligible anyway. And since the light passes through the image, this type of microscope is called a bright field microscope. In essence, what we are saying is that light will be made to pass from under onto a specimen on the stage, which the emitting light will be received by the objective, which will initially enlarge the the specimen, which will now form an image in the tube, which will be further enlarged by the ocular lens and will now be made to be seen as a visual, more enlarged image by the eye, which ordinarily an ordinary eye cannot see. So the diagrammatical representation, as illustrated here, we try to explain what we've just said. The specimen is laid on the stage, adjusted by the mechanical stage, which will be brought to focus, and then the image will be formed, which will be real in between the tube, and then enlarged by the ocular lens, and then diverted to the real eye, where it is now viewed as a more enlarged image. Now, the magnification of a microscope. The term magnification refers to the process of making an object appear larger than it is. It is the apparent increase in size of an object. It has to do with how much your final image is magnified or enlarged than the original object, which in this case is said to be the final magnification. It is denoted by X, and it means how many times it is enlarged. For example, 100 times magnification means that the final image of the specimen will be 100 times bigger than the original specimen. Now, to calculate the final magnification of a compound microscope, the following formula could be used. MT equals to MOB times the MOC. Now, where MT is the total magnification, the MOB is the magnification of the objective lens, while the MOC is the magnification of the ocular lens. In other words, the product of the magnification of the objective lens with that of the ocular lens will give you the total magnification of the specimen under view. And since the magnification of those lenses are given in power, the total magnification will be the product of the powers of objective and ocular lenses, as earlier said. Now, uses of a microscope. Microscopes are used by scientists and healthcare professionals for many purposes, including but not limited to: 1. Diagnosis of infectious diseases. 2. Identification of microorganisms. which are microscopic organisms, and 3, determination of the effect of pathogenic, which are disease-causing microbes, on human cells. And so a microscope is very vital in microbiological laboratory for the purpose earlier illustrated, which is diagnosis, identification, and determination of effect of pathogenic microbes on human cells. If a human cell is infected, the tissue is processed and examined to know the extent of damage or otherwise to the tissues in question. And an ordinary eye cannot be able to view all those details. So the use of a microscope comes to play. Having this preliminary information on the use of a microscope and what is a microscope, I believe at the end of this lecture you should be able to now explain what a microscope is, the principle with which it works, and then why it is used for. In our subsequent lectures, we'll be considering types of microscope and the parts and their individual uses and how to use a microscope, so that as you proceed in the course microbiology, you should be acquainted with this very vital instrument. Thank you.